What can CRISPR actually do for modern agriculture? I've heard statements like it's going to revolutionize agriculture. Now, to me, that statement is a little bit vague. Today, I'm going to give you 7 things that CRISPR can actually do for agriculture. My name is Rahul Pathakar, and I'm going to walk you through this. CRISPR is a pretty easy tool to use compared to previous generation genome editing tools such as Talons. CRISPR is like an automatic nail gun, and Talons were perhaps like a hammer. Both can build the exact same thing, but CRISPR is just a little bit easier to do it with. The first thing on the list is CRISPR gene editing can reduce the government regulation a little bit in comparison to traditional GMOs. This is an ever-changing landscape, but currently the USDA, the FDA, and the EPA all regulate this. The USDA is mostly concerned with whether the plants are safe for other crop plants and other plants in general. The EPA regulates plant pesticides. They're concerned about plants that produce pesticides, or as they call them, plant incorporated protectants. The FDA focuses on the safety and nutritional characteristics of the food and is not concerned about the process where they're produced. The EPA also wants to label new plant varieties. For example, soybean oil that has high oleic acid has to be labeled high oleic acid soybean oil. But one of the biggest changes is the changes in how the USDA regulates these things. The USDA regulates traditional GMOs where everything has to go through approval before it's planted or tested in the field. The Secure Rule establishes exemptions for plants modified by genetic engineering where the modification could otherwise be made through conventional breeding. At the present time, only a single modification qualifies for the exemption because multiple modifications typically result from combining multiple specific traits. The Secure Rule also exempts plants where a given trait's mechanism of action has already been previously ruled by the USDA to be non-regulated. The second thing that CRISPR gene editing can do is it can allow the breeding of linked traits. Traits that are not linked, so for example, traits that are physically not linked, like the traits that are on separate chromosomes or are on distant parts of a chromosome arm, they segregate in Mendelian fashion where you can combine this trait A with the trait B by crossing two organisms that have these traits. However, linked traits can't be bred. So in this example, imagine A is a wonderful yielding gene. If you have this gene, the plants yield wonderfully. And imagine B is a terrible characteristic like the plants shatter like crazy. If these were soybeans, they shatter like crazy. Imagine those two traits are linked. Through traditional breeding, you will not be able to separate those two things. However, with CRISPR gene editing, you would be able to change either of these traits independently without affecting the other. So, for example, you may be able to remove the shattering trait while maintaining the yield. Number 3, CRISPR gene editing can make extremely rare alleles of a gene. For example, they could make a herbicide-tolerant trait in a normal crop plant. Occasionally, weeds can develop herbicide tolerance by random mutations. And they do this by often having the enzyme that is targeted by the herbicide having a mutation that prevents the herbicide from binding properly. When that herbicide can no longer bind to that enzyme and inhibit the enzyme, the plant becomes herbicide tolerant. An example of this is acetolactate synthase gene, where changing a serine 653 to isoleucine confers herbicide tolerance. How would that work? So let's take this example here. If we started with the serine codon of AGT and we wanted to change it to isoleucine, isoleucine can be any of these 3 codons. So the simplest change we could make is we could change that G to a T. However, the chance of getting that exact change to happen randomly is so rare, it's going to be really hard to find it. And in fact, known mutagens like EMS that is commonly used in breeding cannot make that change. They can make the GC base pair turn into an AT base pair. So they can basically make a G turn into an A, and that's not useful for this case here. Number 4,
You can recreate an existing gene allele in a new breeding line. Here perhaps you have a corn line that has a very nice high-yielding trait. And you have another line that has other desirable traits. Maybe it's drought tolerant. With CRISPR gene editing, you can transfer the DNA information from line 1 to line 2. So you'll be able to maintain all of the characteristics of line 2 with the exception that you've added in this high-yielding trait. Number 5, you can knock out multiple genes all at once. For example, you may have a gene family that regulates a trait. So knocking out one gene that governs that trait would not be enough to confer the trait. Rather, you may have to knock out several traits. Like in this example here, maybe there's 3 traits that need to be knocked out. This can easily be done by doing multiplex CRISPR gene editing, where we have multiple guide RNAs that target all the genes that you want. This here is a way of doing 6, but people have done more than 12 of these already, all at once. Importantly, the USDA does not exempt multiplex edits straight away. However, under the old system of MI regulated, they actually would approve some multiple edits. So it should be possible, but with a little bit of government oversight. Number 6, you can create breeding partners that can allow for quick trait stacking. For example, if you had a good tasting corn line and you had 3 different high yielding corn lines, you might cross them together in traditional breeding. And then you would try to get the good taste along with this high yielding trait. Or you would get the good taste with this high yielding trait, and so on. With gene editing, you could take the good tasting trait and put it into each one of these lines without changing them in any way other than the good taste. In this scheme, then you would have good tasting, high yielding line A, B, and C that you could then cross together to make even higher yielding good tasting lines in a quicker fashion than you could with traditional breeding. Basically, you could stack the traits faster than with traditional breeding. Number 7, you can transfer trait knowledge from a model plant to crops. There's lots of valuable knowledge that comes from model plants like Arabidopsis, where we know a lot of gene function. You can take that knowledge and then you can try to use that knowledge to recreate the trait in a more valuable plant like corn by moving that trait over. So here are 7 actual things that you can do with CRISPR gene editing to help agriculture. If there are other things that you think should be on this list, leave them in the comments down below. You could also stop by pathogar.com if you want to get more science tips and protocols. Thank you for watching, and you might want to check out these videos next.